All right, so this is video 3. We're looking at cellular surface modifications. Again, for my class, as we're studying out of the Hole's 16th edition, the PowerPoints provided by the publisher are going to be slides 26 and 29. Um, it's a pretty short video. There's only a certain number of these we look at. The very first one we look at is a surface modification called cilia. Now, cilia basically on the surface of cells, we're going to have hair-like extensions that come on here. And what they do is, and we looked at this in one of our types of tissue in the histology lab, we called it the pseudostratified columnar epithelium, which is lining your respiratory tract, has cilia on the surface of it. And so as I'm sitting here, which I probably don't need to be doing, drawing all these little things on there, what it forms is this little layer, and it almost would be the equivalent of having astroturf on the surface of the cell, but we can control each one of those little grass-like extensions that come off of it. And so what they do is if I have something that gets on the surface, right, so let's say I'm in the south in the wonderful spring when the pollen goes crazy and I get this pollen, my throat will become irritated and I'll start producing a lot of mucus because in this area we had what we called goblet cells that produce this mucus out here that'll trap it, and then these little guys will just take it and move it away, kind of get rid of it, so that we can either cough it out or swallow it, which might sound gross, but that's one of the ways our body gets rid of things like that. Another area where this is going to be is in the fallopian tube. When the oocyte is released from the ovary, it gets into the fallopian tube. The fallopian tube is lined with cells that have cilia on it, and they will move back and forth, kind of like a crowd surfer, trying to move that egg or oocyte towards the uterus. So that is cilia. Another type is flagella. Now, where cilia is going to have, like in cilia, each cell is going to have a lot of these little extensions. The flagella, there's only one, and it's only found in one type of cell. It's called a sperm, and so you got the little body of the sperm, and then you're going to have this long tail that's designed to whip back and forth to try and get that sucker moving in one direction. And so the sperm is the only cell that has a flagellum on it, or a flagella, and it only has one. And that's the difference between cilia and a flagella. Now on this, you see the pictures of it. It shows how it's moving things along. Now another one that kind of is easy to confuse with cilia is microvilli. Now back in again, the histology lab, we looked at this type of tissue called simple columnar, and it was in the digestive tract we looked at it. And so our picture, which again, this is a bad picture as I'm trying to draw with a little mouse on this. So we talked about how these little areas that stick into the digestive tube are called villi. They are lined with simple columnar epithelium. Now, if I was to take a little cross section of that little area and blow it up and look at the cells that are on there, again, they're going to be simple columnar. We're going to, again, in the book world, it seems easy to label. There's one cell layer thick and they're column-like cells, but we know in the real world it looks a little different. But if I could actually go in, and we'll see a picture of this again in the next thing, but I just kind of like messing with this, trying my best to draw it. If I was able to really focus in on this simple columnar cell, I would see that at the top, that the cell membrane is actually very wavy. It goes back and forth kind of like, and when I draw it, it ends up looking like a Bart Simpson kind of. But these little waves on the top are called microvilli. And again, it is all about increasing surface area. Anatomy and physiology, what something looks like is determined by what it does. And we need to increase the surface area because this is for absorption. And so we have the macrovilli, or just villi, that are these finger-like extensions into the tube we call the GI tract. And then those extensions are lined with simple columnar cells, which on their surface have these microscopic villi that stick up. Now, in between, the difference between this and the cilia is not going to have any cytoplasm in it. It is like a projectile. It is a little protein kind of like filament that sticks out. Where these actually, if I was to able to really microscope, magnify my image in, you'll see that cytoplasm is all up in here. So it's helping to produce a surface area. Now, this, I am not worried about granules or inclusions. Um, this is going to be different things. We'll look at it mainly in melanocytes, how they'll have inclusions that are like little arms that go through the cells to help provide another type of surface area. But again, I am not worried about inclusions. The microvilli, cilia, and flagella are the only ones that I'm worried about. And then this picture, which we will end with, is the picture, again, from the book, showing these microvilli. And again, there is cytoplasm that goes into here. So it's increasing the surface area, unlike the cilia, which is just extensions. And I hope that makes sense.